Hey guys, today we're going to talk about our first topic in Unit 4, which is Meiosis, Genetics, and Heredity. Part 1, we're going to look at the process of meiosis and how it keeps its chromosome numbers constant as it undergoes this process, and also how it leads to increased variation in species. So to begin with, there's quite a bit of vocabulary we need to cover here. You can see all kinds of different words highlighted in red. Those are going to be really important vocab terms. I'll kind of breeze through them, and we'll practice those in class a bit more. To begin with, we really need to understand the process of meiosis is the creation of sex cells. When we talk about sex cells, there's going to be a term you're going to see in here further down. Gametes, that's the term we'll use in class, the creation of gametes. What are those gametes? You can see them over here in this picture. We have a sperm cells headed towards this egg cell. Those are your gametes. Those are two very special types of cells that are created to lead through the process of reproducing a species. So what are the major types? Well, so far we've talked about just regular types of body cells, and we haven't really given them a special name in class. The special name that we're giving them is called a somatic cell. All right. A somatic cell is a body cell. Those would be the things created by mitosis. We've already talked about that. Those would be things like if you're you need to replace some skin cells, those skin cells are going to be regenerated into more skin cells, and in order to do that, mitosis has to happen. The cell doubles the DNA, and then it divides once and creates these cells are diploid. Diploid is a term we haven't yet talked about. We abbreviate it as 2N. This means we have two sets of chromosomes, one from the father and one from the mother. You're going to see why that's important, because gametes are haploid. So we create the sperm and the egg cells through the process of meiosis. We see haploid cells created. Those are symbolized just as N. Haploid cells only have one set of chromosomes, just one set. And that makes sense if you think about it. The sperm cell has half the required DNA of a human. The egg cell has the other half required DNA of a human. When those combine and fertilize and form a zygote, then we start creating one human with the appropriate number of chromosomes. So that really makes sense. When we start talking about chromosomes, we want to refer to them as either autosomes or sex chromosomes. You'll see that becomes important. Autosomes are really chromosome pairs numbers 1 through 22. You'll see what I'm talking about in just a second. And sex chromosomes are the 23rd pair, written as X and Y. Next up, we can see these diagrams. These diagrams are of chromosomes. One is before replication and one is after replication. So before replication is here. There's one pair per type of chromosome. So for chromosome 1, these are in a these karyotypes are these diagrams, and they just basically organize chromosomes based on size and shape. So this is one pair per type, one copy from each parent. What happens is DNA replicates before any type of division, and then we end up with two copies from each parent. So you can kind of see the difference here. There's basically one X-shaped homologous pair, and the other one as well, which all in all is double what's on this side. So really, replication, we've talked about that, it's just doubling DNA, getting ready for any kind of cell division. So meiosis is a creation of these gametes. We've covered that. It really happens in two rounds. The first round is referred to as meiosis 1, and then the second round we're going to call meiosis 2. We'll get there in a second. All of the phases are just the same names as they were in mitosis. What happens in them is different, but the names are the same, and so it's similar in that regard. So remembering the stages, the PMAT, prophase, metaphase, anaphase, telophase is a pretty simple process to kind of review. What I wanted to highlight in meiosis with you guys was really prophase 1. I think if there's any phase that we're going to talk about in this class, specifically prophase 1 is probably 90% of the time what's going to happen. There's a lot of stuff that's happening in prophase 1 down here. You're going to see us talk about that a lot. homologous chromosome pairs are going to line up next to each other, or they're going to get next to each other. They're going to undergo this process of crossing over. They're going to exchange genetic material. All of this stuff is creating more diversity in an organism. It's making different combinations of the DNA possible to get into these other cells. We can go to the process. The next process here is metaphase. Stuff goes in the middle just like it always did. Instead of getting one long line, the homologous pairs go next to each other. So you can kind of see this happening here. They kind of pair next to each other rather than get in like a single file line down the middle. And then instead of getting ripped apart, one pair goes one way, one pair goes the other way into the other cell, into the end of the cell in anaphase. So moving on to meiosis 2, the end of meiosis 1 and meiosis 2. Looking at meiosis 2, we're going to start with prophase again, except instead of prophase 1, we call it prophase 2. At this point, the homologous pairs are already going back to the middle, or they're getting ready to go back to the middle of metaphase, and then they're going to be separated apart into individual sister chromatids, and then finally pulled into individual cells that are haploid. So we can see that these two cells are formed in this intermediate kind of stage here to start meiosis 2. Then we see homologous pairs in the middle, so moving this way, and then we see the sister chromatids get pulled apart, and then one sister chromatid goes to one side, one goes to the other side in telophase 2, and we end up with 4 separate haploid cells. All right, so that's really important there. 4 separate haploid cells. So some more key vocabulary, and we kind of covered a little bit of this going through, but I know it's fast. Homologous pairs. Homologous pairs are chromosomes that are the same size and shape, and carry the same type of genes and traits and those kind of things. A tetrad is a group of 4 chromosomes, 2 sets of homologous pairs together. So you can see in prophase, we just talked about prophase here. Prophase 1, we can see right here is our tetrad being formed. It's just the 2 homologous pairs hanging out together. Crossing over occurs when we have tetrads formed in prophase. That's an exchange of genetic material of non-sister chromatids. And this diagram down here shows you that. When we have homologous pairs of chromosomes, right here, these are homologous pairs. When the homologous pairs hang out together, they form this tetrad. When they form a tetrad, then crossing over occurs, and then this is the result here. So when we cross over, that's the result, is exchanged genetic material. So we can see that this chromosome, and they're trying to make it color-coded for you easy, this red chromosome pair and this yellow chromosome pair cross over, and it looks like they're, the bottom parts just kind of swap. So now there's not a whole red one and a whole yellow one. That shows that genetic material has exchanged, genes have kind of crossed, and creates a unique combination here. And then a dyad, you saw that kind of in anaphase 2. We saw the dyads get pulled apart. Those are really the sister chromatids when they're finally separated. We talk about as a dyad. Mitosis flowchart and meiosis. Part of what we're going to do in this class is compare mitosis to meiosis. So interphase is what cells are doing, normally growth and development, that kind of thing. So when they're in interphase here, in order to head to mitosis, remember what has to happen? Well, they have to double their DNA. So they replicate, put ×2 here, and move this way. When we move this way, you can see we had 46 individuals. We were a 2N diploid cell, right? And then we doubled, and now we're 4N technically. There's just 46 chromosome pairs. There's a lot of DNA there. What happened in mitosis? We split back into 2 diploid cells. Again, we want to be diploid, right? And we want them to be the same. So we use that example of our skin cells. We need to replace. Well, if we need to do that, then we need to make sure that we double our DNA, push it into 2 cells, and split. So that's our flow chart of chromosome number. Diploid cells to 2 daughter cells that are identical and diploid. In meiosis, in meiosis 1 and 2, we start with their same diploid cell, and then we double our DNA in replication. So we have this 4N thing here, and then we start our cell division. Instead of one round, we're going to end up having 2. So now we have 23 chromosome pairs in each cell here, and then we start meiosis 2, and then we see 23 individual chromosomes. So we have these pairs, and then we split the pairs up again, and those pairs go into each individual gamete, and all of these are sex cells down here. These N or haploid cells are here. So they're haploid sperm or egg cells. What I'm going to ask you guys to do here is take a moment. You're going to probably need to pause this thing, and I want you to write down 3. So I want you to write down 3 that you think you're going to remember here, and I'll put it up here on the board. Know 3 of these. That's what I want you to do. So there's 5 up here, 3 you can remember. We'll go over them all right now. DNA replication, remember, it occurs during interphase before mitosis begins. And in meiosis occurs during interphase before meiosis begins. So interphase is where DNA doubles. This is the same kind of thing, right? So similarity. Number of divisions, 1 and 2. This is definitely going to be a difference. Synapsis of homologous chromosomes crossing over. This is going to be a difference. Number of daughter cells, 2N, another difference. N, creating 4 cells, creating 2 cells, definitely another difference. And then role in the human body, this is going to produce our somatic cells, our body cells, and this produces gametes. Remember, somatic, we'll use the vocabulary we talked about, somatic cells. And then just some diagram here to look at the difference again, just basically what we just went over. What I did want to highlight to you is a project that we'll be doing in class, or we may do in class, dealing with the different phases. This diagram may be helpful. You might want to refer back to it. It talks about the phases here and what's happening. Those are key things that are happening that you'll need to know.